Okay, good afternoon, class. How are you today? I hope you are all doing well. Did you study our lesson? Did you study our lesson? Okay, let's start. Okay, our topic for today is about mitosis and meiosis. Okay, let's start with the learning objectives. Our learning objectives is to differentiate mitosis and meiosis. Second, identify the different phases involved. Third, determine the key events in each stages of cell cycle, both in mitosis and meiosis. Fourth is to explain how cell division functions in reproduction, growth, and repair. Describe the chromosome number changes throughout the life cycle. Okay, who can give me the definition of mitosis? What is mitosis? Anyone? Okay, good. Okay, what is mitosis? Mitosis comes from the Greek word mythos, which means thread. It is a nuclear division plus cytokinesis and produces two identical daughter cells during prophase, metaphase, anaphase, and telophase. It is also part of the division process in the context of the cell cycle, in which the DNA of the cell nucleus is split into two equal sets of chromosomes. It is also each daughter cells gets a perfect full set of chromosomes in all of these cases, because they don't usually function when they are too few or if they are too many. So that's why they are divided equally. Okay, mitosis has stages. Okay, the first phase is the interphase. Interphase is the phase of cell cycle in which a typical cell spends most of its life. During this phase, the cell copies its DNA in preparation for mitosis. The nucleus is clearly visible with one or more distinct nucleoli, and the chromosomes appear as irregular granular form, thus cannot be recognized. Okay, in this interphase, it has three subdivisions. First is the first growth period, or the G1. Second is the synthesis period, or the S phase. And the third is growth period, second growth period, or the G2. Okay, let's see here. During this first growth, here you see the G1 phase. This is the period before the synthesis of the DNA. Okay, and then the S phase here is this is the phase during which DNA synthesis takes place. And then here the G2 phase, or the second growth period. This is the phase between the end of the DNA synthesis and the beginning of the prophase. Okay. And then that's the mitosis begins here. That's when the mitosis begins after the G2 phase. Okay, next. Here. The interphase, the nucleolus, and the nuclear envelope are distinct, and the chromosomes are in the form of thread-like chromatin. Okay, you see the picture, the image. I hope you can identify. Okay, next phase is the prophase. During mitosis, prophase is when the chromatin condenses into two rod-shaped structures called chromosomes, in which the chromatin become visible. Okay. This process, known as chromatin condensation, is involved with the condensing complex. It follows immediately the S phase and the G2 phase of the cycle in the interphase. It marks by the condensation of the genetic material. Okay. In the prophase, it includes the nucleolus disappears, the chromatin condenses into chromosomes, the separation of introsomes, and the formation of the meiotic spindle. Okay, it includes also the prometaphase. Let's discuss this next time. Okay, next is the metaphase. Metaphase, double-stranded chromosomes are aligned at the equator of the cell. Mitotic apparatus completely formed, and the nuclear membrane completely formed. In this stage, the microtubules start pulling the chromosomes with equal force, and the chromosomes ends up in the middle of the cell. They ends up in the equator line in the metaphase. Next is, okay, here you will see that the metaphase thick coiled chromosomes are lined up in the center of the cell in the metaphase plate. Spindle fibers are attached to the chromosomes. They are aligned in the equator line. Okay, the chromosomes are aligned in the equator line. That's the chromosomes. Okay. Next is the anaphase. The anaphase, the phase of migration, centromeres of each chromosome divide, and then here the cytokinesis begins. Okay, we will discuss the cytokinesis later. And here is the presentation of what anaphase is. The chromosomes break at centromeres, and sister chromatids move to opposite ends of the cell. This is the sister chromatids. Okay, this one are the sister chromatids. The splitting of the sister chromatids marks the onset of the anaphase. Okay, the sister chromatids becomes the chromosomes of the daughter nuclei. Okay, next. Okay, are you okay, class? Did you understand? Okay. Okay, next. Okay, next is the telophase. The telophase, the phase of migration. Centrioles are replicated. Chromosomes begin longer, thinner, and distinct. Nucleolus reappears and new nuclear membrane forms. Mitotic apparatus disappears. Okay, here is an image of how the telophase occurs. The telophase, the chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming. The cytoplasm may be dividing, okay, resulting into daughter cells. They are called haploid. Okay, this one, the daughter cells, they are called haploid. Each of them only has 23 chromosomes. One set of these consists of 23 chromosomes. Okay, did you get it? Okay, next. Okay, this is what cytokinesis is. Each round has a cytokinesis. Cytokinesis is the marks that the process is already completed. Cytokinesis, the division of cytoplasm that results into new daughter cells. Okay, we have different process in plants and in cell cycle. Okay, here I hope you get the process of the mitosis, the interphase, where the cell spends more of its life in this phase, and then the prophase, the chromosomes paralyzes, the metaphase, they are aligned in the equator line, the anaphase, the sister chromatids here are splitting already, and then in the telophase they are forming two different cells. Okay, they are with haploid with 23 chromosomes each. Okay, next. Okay, did you understand what mitosis is? So we can move on to our next topic. Do you have any question? Okay, let's begin with meiosis. What is meiosis? Meiosis is a process that produces 4 daughter cells with haploid number of chromosomes as the parent cell. It involves 2 successive division, meiosis 1 and meiosis 2, occurring with gametogenesis. Okay, what is meiosis 1? It is the result in reducing the number of chromosomes. During the let's go back. During the meiosis, the genetic material of a diploid germ cell undergoes 2 nuclear division and resulting into 4 haploid daughter cells. Each daughter cell has 1/2 of the number of chromosomes as parent cell. Okay, we have 2 stages of the meiosis. Let's start with the meiosis 1 and its phases. The phases of the meiosis 1, the prophase 1. What is prophase 1? Who can read about the prophase 1? But before the dividing cells enters meiosis, it undergoes first in a period of growth called the interphase. Okay, the prophase 1 occupies the longest duration of the meiosis. It is subdivided into 5 subphases: the leptonema, the zygonema, phakinema, diplonema, and the diakinesis. Okay, we will discuss this next time because we are lacking out of time. Okay, you kindly study in advance so that you can participate in our next class. Okay, let's move on to the metaphase 1, which is part of the meiosis 1. The metaphase 1 is the synapse homologous chromosomes are aligned at the equator of the cell. It's like in the mitosis, they are also aligned in the equator cell. The anaphase 1, the whole chromosomes from each tetrad separate and migrate toward the opposite poles of the cells. Okay, the telophase 1, meiosis 1, ends when the chromosome of each pairs arrive at the opposing poles of the cell. We have here the cytokinesis that that says that mark that the process is already completed. Okay, cytokinesis completed, resulting into two daughter cells with haploid number of chromosomes. Okay, you can study the diagram so that you can participate on our next class. Okay, let's move on to meiosis 2. Meiosis 2 follows interkinesis, no chromosomes replication between meiosis 1 and meiosis 2. Okay, in this division is like mitosis, the number of the chromosomes does not get reduced. Okay, we also have phases of meiosis 2. It don't have a anaphase. Okay, the prophase 2, chromatin condensed to form visible chromosomes again. Yeah. The chromatin condensed to form visible chromosomes again. Okay. The metaphase 2, the chromosomes align at the equator plane, which is rotated 90 degrees compared to equatorial plane in meiosis 1. Okay. The anaphase 2, the spindle fibers connected to each chromatid shorten, pulling one sister chromatid each pole. And then the last is the telophase 2. Meiosis 2 ends when the sister chromosomes have reached opposing poles of the cells, and it is also ended with a cytokinesis. Okay, don't forget that cytokinesis marks the end of the process. Okay, that's the process of the meiosis 2, the prophase 1, the chromatin condenses. And then in metaphase 2, they are aligned in the chromosomes aligned in the equator line. And the anaphase 2 naman is the sister chromatid is pulling apart. Okay, and then the telophase forms a sister chromosomes, okay, opposing poles of the cells. Okay, and then the cytokinesis marks the end of the process. Okay, here is another image of how meiosis 2 looks like. Okay, that's end our lesson for today. I hope you understand our topic, and please study your lesson. Tomorrow I will give a quiz. Okay, goodbye class. Thank you for listening.